60th anniversary. It's Paul Kennedy in Toronto doing the interview with the laureate, Dr. David Baltimore in California. And my first question, once again, what got you interested in science? How did you first get interested in what you're doing now?
When I was in elementary and high school, I discovered I did mathematics and science well. I enjoyed it. And I didn't realize then, but I think what was the truth was that I see the world from a scientific perspective. And so being able to understand the world the way science allows us was to me very satisfying. And my mother picked up on that and reinforced it. She was a scientist and sort of helped along with my development as a scientist.
I love the fact that you see the world from a scientific perspective, as though you put glasses on and it changes the way you look at the world.
Well, I don't know that I put glasses on, but it just came naturally.
What is it that makes you a scientist then? How do you look at the world that makes you different from an artist or a construction worker?
Well, your mention of an artist is interesting to me because I've just written and will soon publish a little piece on the difference between art and science. And one simple way to see that is that a scientist is restricted in what he or she can think about by the external reality of the world. And an artist isn't. An artist has no boundaries for what he or she can think about. And that's a very fundamental difference. And it means that a scientist is constantly looking for a formulation of the reality of the external world that makes sense, that is consistent, and that can be tested and found to be an appropriate description of how the world works.
Is there a similarity maybe in what's called often the Eureka moment, the moment when you come to a realization that everything does sort of match, that the world has been explained the way that you described it in the first place?
Well, yes. If you're functioning as a scientist, you look for that moment when clarity comes to a situation and you can suddenly think about it in logical terms. It isn't amazing. That's a great moment for a scientist.
I can only imagine. Almost artistically, I'd have to imagine it. I'm also curious about how that urge towards science was nurtured. You say your mother was a scientist, and did you have teachers who were scientists?
I had some teachers who were scientists, and particularly as I got into graduate work, I had people who were very important to me in guiding my development. And it was really a continuum from the home I grew up in to the schools that I went to the people I worked with.
Why biology? Why were you interested in that particular branch of science, not physics or mathematics?
I don't actually know why I got into biology, but partly it was that I could think about biology, and I was never good at thinking about physics, particularly as physics was developing in the 20th century. And so it didn't give me that satisfaction that biology did. But it was also that biology was an open door. I started thinking about experimental science in the 1950s, and that was just after the structure of DNA was discovered by Watson and Crick. And that discovery completely changed how we thought about biology, because biology became an information science. It became a science built on how information was passed from one generation to the next by evolution. And a very complicated mechanics of transcribing and translating the information in DNA was just beginning to be understood. And so I was in a situation where almost anything I did in biology was new and interesting. And that was, again, very fulfilling, very exciting, and enabled me to rapidly go from being a student to being a leader in the field.
You mentioned Watson and Crick. How aware were you at that moment of what they were doing?
I was not, in 1953, when they published the paper, I was still in, I guess, just getting into high school. So I don't think I was aware of it at all. But by the time I went to college, which was 1956, I began to become aware that there was this molecular way of looking at biology, dubbed molecular biology, that provided an understanding in a depth that had never been there before. Biology was so largely an observational science until that point. And so I sought out books, journals, people who could help me understand what was going on, and spent particularly the latter part of my undergraduate years doing that. Interestingly, I followed in the footsteps of Howard Timon, who had gone to the same school I did, Swarthmore College, but had gone there 4 or 5 years earlier. And he also discovered that there was a new way to look at biology, and devoured every piece of information he could get that positioned him to begin to become the great experimentalist that he was.
Fascinating. They must have had something in the school water. I go back to what you said about art and science. Biology and the kind of mathematics you're talking about were also considered to be different worlds. That the fact that you could ultimately, I guess, it led to the genome, to mapping the genome, but to make something, that kind of science, which was another kind of science, was a really exciting time. And you, as you say, got there right at the crest of the wave.
Yeah, although I learned some mathematics, I even minored in mathematics, I didn't apply much mathematics to the work that I was doing in any direct way. And the interesting thing is that today, a young scientist growing up has to think more mathematically, and is enabled in doing that by the new thinking in biology that's constantly emerging.
I understand that when you were still quite a young man, I think even in high school, you got involved really in what's called real science. You were involved because of some of your teachers and experiments and working on things that was way beyond what was the normal case.
Yeah, I was lucky that my mother, who was at that point teaching at the New School for Social Research in New York, saw on the bulletin board an announcement of a summer program for high school students and brought it home and said, would you be interested? And I said yes, and so I applied to this program at the Jackson Laboratory in Bar Harbor, Maine, was accepted and spent that summer between my junior and senior years of high school working with some of the greatest scientists in biology at that time, who also spent their summers at the Jackson Lab, and some of them worked there full time.
It must have been very empowering. Like you say, you saw the world from a scientific perspective, and now you got excited with that kind of excitement about biology. You must have felt that the world was your oyster.
Yeah, that's. I wasn't much conscious of the rest of the world, but it certainly was exciting, satisfying, and shaping of the rest of my career.
Did you know that at the time?
No, I wasn't that self-aware. I was sort of, I think most kids are, sort of surfing along on the waves that came by.
I like to follow the career of brilliant people like you in how much they knew about what they were doing when they were doing it. And so even that kind of decision, the trajectory of your life right now follows a route that has explanations, almost scientific explanations. Your mother was an influence, your teachers were an influence, Bar Harbor was an influence, and yet it all now sounds logical in a direct line. It wasn't necessarily, though, at the time.
That's right. I'm now 80 years old, and that has me thinking back to the trajectory of my life, and I can see it falling into an ordered pattern, just as you've described. But in living it, it didn't seem that way. And I think that's probably a good thing. I think too much self-awareness maybe inhibits your ability to be creative because you keep thinking about yourself. I think that's also maybe a difference between artists and scientists.
It's an interesting distinction anyway, and one we could do. Another, this is a truism that often follows the Gairdner Foundation, that when you win a Gairdner, you're going to win a Nobel Prize. So let's go find out with the Gairdner first. Do you remember how you found out that you'd won a Gairdner?
Quite honestly, I don't.
Okay, honest answer is good. Did you come to Toronto to accept it?
I did come to Toronto. It might even have been the first time I was ever in Canada. And no, probably not. I had been in Canada before. I knew Lou Siminovich, who was in Toronto at Sick Kids, and knew some other Canadian scientists.
Maybe we should go into what you were awarded for. I mean, the science that you did that won this. And I guess we should find out why not only you chose biology, but why you got interested in viruses.
Well, I got interested in viruses because as I entered experimental biology, first of all, there was a tradition of working with viruses that was largely focused around viruses that infected bacteria. And many of the advances in molecular biology that had been made over the previous decades came from work with viruses. So that tradition was well established. But I didn't really want to work with bacterial viruses because I was interested in humans and in mice And so I got to wondering whether you could do the same kinds of things with viruses that infect animals. And I spent the first year of my graduate career thinking about that question, and finally got the advice from my effective mentor, Salvador Luria, that I should give it a try and see what it looked like. And so he helped me find somebody to work with over the following summer, and a course at Cold Spring Harbor that had just started on animal viruses. And after that introduction, I realized that this was a field that was just on the takeoff platform. And so I sort of readjusted my life to work on animal viruses and to begin to apply some of the biochemistry and molecular thinking that was going on to these very interesting viruses, many of which had as their genetic material RNA, while the rest of the world has DNA as its genetic material. And so they presented a unique set of very interesting problems.
We're sort of trying to follow your trajectory, and I'm also curious about how you went from first science to biology, then biology to viruses, and then viruses to retroviruses. How did that happen?
Well, so I began to work on RNA-containing viruses in 1961, really. I had moved at that point to be a graduate student at the Rockefeller University, working with an animal virologist. And the first virus that we worked with was a close relative of poliovirus. And I did that for 8 years. And polio was a very interesting example because it's a very simple virus. And so we could learn a lot about it, and a large percentage of what made it tick. And somewhere in about 1968, I decided to branch out a little bit and look at some other RNA viruses, in particular focused on a virus that my wife worked on called vesicular stomatitis virus. She was at that point in my lab, later a professor at Harvard. And so we started looking at this virus called VSV, or vesicular stomatitis virus, and realized that it completely changed the way we thought about genetic systems of viruses, because poliovirus carried in its particle what we can call sensible RNA, RNA that encodes protein directly. And vesicular stomatitis virus carried exactly the opposite strand of RNA, one that was senseless, but that if you copied it, produced a sense molecule. And that was a revelation. And that allowed us to effectively understand how most viruses worked. They either worked like polio or they worked like VSV. But there was one class of viruses that didn't seem to fit that picture. And that was the viruses that cause cancer. So it was a very important class of viruses because it potentially held the secret of cancer if we could understand those viruses. And so I started working on them. And the first experiment that I did was to look for the ability of the virus particle to make either RNA or DNA. And I didn't know which it would be. But they're RNA viruses, so the paradigm that we had would have suggested it would make RNA. But Howard Temin had suggested maybe 10 years before that maybe they made DNA. And so it was up in the air. And I began to apply biochemical methods to these virus particles and very rapidly showed that they made DNA and that there was a system in the virus that copied the RNA genome into DNA. That was revolutionary.
5 years for the Nobel, 4 years for the Gairdner. How did it feel to win the Gairdner? Was it a stepping stone?
Well, it certainly alerted me to the fact that we might win the Nobel Prize. But it was at the time an enormous honor to follow in the footsteps of people who had gotten the Gairdner Award, who were some of my heroes in science. And so I began to realize from that I was entering a new world of the most honored scientists working in biology, and that was humbling.
Do you remember what it was like in Toronto? Do you remember a ceremony, or do you remember being showed around? What do you remember about that time? You were a young man. You were 36 years old, I think, when you won the Gairdner, 37 for the Nobel. Does a young man deal with that kind of awards, honors, being paid tribute?
You know, in retrospect, I was very young. Most people who get awards like this are significantly older. But 36, 37 years old was not a young man to me at the time. We used to say that if you're over 30, you've entered the realm of irrelevance, and that it's really people in their 20s who make a difference in the world. So I didn't think of myself as a particularly young man, but in retrospect, I certainly was.
How important are these prizes to a scientist in early career, mid-career, or late career, I guess? I mean, what does it change?
It certainly provides a validation of the work that you've been doing since you started as an experimentalist, since I started as an experimentalist. And that validation gives you a sense that the world takes you seriously, and it is a little bit liberating because you don't feel like you have to follow the trajectories opened by other people. You can make your own pathways. I was a moderately arrogant young man, so I was already doing that to some extent, but it certainly was a validation. It also changes your life in very mundane ways. You get lots of invitations to travel, to go to conferences, to provide advice to people. And so now you have to control your life with a little more understanding of what matters to you. And I realized that what mattered to me was doing science, and not getting awards.
It sounds to me as though you're being almost artistic in those kind of choices. I mean, you're following your passions. You followed your passions as a young child, even to get interested in science, and then all the direction since then have been not necessarily made because it was a smart decision or a practical decision, but because something you loved was there.
Well, that's in fact true. I was following my passions. That's a bit of a cliche these days, but it certainly defined how I sort of constructed my world. The other thing that I began to take an interest in, that was more like in 1980, was the institutions of science. And I took on administrative roles, ultimately becoming president of Caltech, as a part of my interest in science, because science is done in institutions. And unless institutions are strong, they can't be the home for effective science. And so I saw administration as a continuum with the experimental work that I was doing, and actually never stopped doing experimental work.
That's a very rich answer for two reasons. One, institutions are also places where older scientists rub shoulders with younger scientists. And there's a mentoring process and a teaching process too. And is that important to you as a scientist to deal with the next generation and the generation after that and inspire them to go further?
Absolutely. Your responsibility as a scientist is not only to try to understand the complexities of the world, but to prepare the next generation to take that over.
Do you have an example of how you've done that? Can you talk about another scientist you've worked with, or a class or something where you really felt that was happening?
Well, I ran a pretty big laboratory for most of my career, maybe 20, 30 people working with me. Most of these were trainees who would go on in science themselves. And so I've trained some 250 scientists, and they populate the science of today. We just had a celebration of my 80th birthday, and there were about 400 people there, most of whom had worked with me in one way or another over the time I've been in science. Some are now university presidents. Many are honored professors, members of the National Academy. So training the next generation has been a seamless part of my life ever since I began in science.
This is an interesting time to be a scientist because science isn't necessarily valued around the world or in certain places in the world like it used to be. How do you feel about that?
It's true that science is not valued by the general public as much as it might have been years ago. Actually, I think it's an ambiguous relationship because, in fact, scientists or science is such an important part of the modern world. And all of us, with our gadgets and with our medical treatments, are taking advantage of new science continually, driven ahead in the way we live by science. And so for people to be unappreciative of science seems odd at this time in the world. But maybe it's because the science of the world is moving people ahead faster than they would like. And I think that that's a little bit of a more than a little bit of an issue.
Given that, though, you have no trouble advising people to follow a career like yours. You would encourage young people to become scientists.
I would absolutely encourage young people, and it's largely because I myself have been so enriched and satisfied as a human being by my life in science that I encourage other people to take that on. If they were born with a sort of science-based way of thinking.
When you look back on your career, would you change anything?
Not much. I think the decisions I've made along the way have been good ones. Perhaps the most difficult one was moving to California, because I spent most of my career, all of the early part of my career, on the East Coast. I was born on the East Coast, born around New York. And so I never imagined myself living in California. But the opportunity to be president of Caltech was such an attractive idea, because it's one of the unique institutions of the world, that I came to California. And even though I've stepped down as president, my wife and I have stayed in California because it's such a wonderful place to live. And Caltech is such a wonderful place to be a faculty member.
You mentioned institutions in science and their importance. Yeah. I was wondering if you'd like to reflect for a moment on the Gairdner's place in that, the Gairdner Foundation.
Oh. Well, an award like the Gairdner Award, which has been, in a sense, validated by the receipt of the award by so many superb scientists, acts to provide a sort of quality control over the scientific enterprise, and something for people to aspire to, not because it's the right thing to aspire to awards, I don't think it is, but because achieving recognition of that sort gives you a sense that the decisions you've made in life were, in fact, the right ones. And not everybody, in fact, probably not many people in their lives get that kind of feedback and validation. So the very hard work that the Gairdner people do to decide every year who should get this award. And that's, I know, a long and arduous road before it settles down on just a very few people. And that's all extremely worthwhile, because it provides that validation. And so I think, particularly because it's a distinctively Canadian award, it's valuable to the whole Canadian population in helping to define what is good science, what is worthwhile supporting, and what's going to change their lives.
I think this has been great. You've given me wonderful answers here. I wonder if you could do one thing for me, and that is try to tell me, or tell the audience, in a minute or so, what you got the award for. What was it what was the science that you did that earned you a Gairdner and ultimately a Nobel Prize?
It was to bring a new understanding to science that hadn't been there before, of just the way the informational molecules that drive biological systems, that drive development, that drive behavior, just how those informational molecules relate to one another and how they collaborate to allow the systems to function. So that was one very important outcome of the work we did. But the other was in relation to cancer. Because cancer at the time, in 1970, when we did that work, and I might say I did the work personally in my lab, cancer was an enigma. We didn't know why cancer cells behaved so differently from normal cells. Didn't listen to the signals that normal cells listen to. And didn't obey those signals, and so grew out of control, which is the hallmark of cancer. And by providing a key piece of information, we were able to illuminate the nature of cancer and direct the work of the next, well, it's been now 50 years, to bring us to where we are today, which is that we are beginning to really control cancer, and we are seeing cures of cancer that we were never seeing before. That's not due to my work necessarily, but to the work of the whole field. But we gave it a push in the right direction.